Hi, we are iGEM Qdai, and our project for this year is Alternative to Canary. We were the first team from Kyushu University to participate in iGEM last year. So our history in this competition is still young. Nevertheless, we were able to proceed with our experiments thanks to the support and feedback given by many experts. Without further ado, let's get started. We aim to create an alternative to the canary in the coal mine. That is, canaries used to be utilized as breathing detectors of toxic gases to ensure the personal safety of miners. However, as time went on, their use decreased since new electromagnetic devices such as gas detectors and robots came into play. It seems to be a suitable solution, yet they still have a couple of problems such as the use of platinum, which is a degradable resource, the dependence on electricity, the high production cost, and so on. In response to these problems, we've been working to create a composite gas detector using evil bacteria, which is inexpensive and does not require electricity power supply.
We decided to create a device to replace the canary. This device is a gas detector that applies sensitive biology to warn of the presence of carbon monoxide. The device is expected to be used not only in coal mines but also in farms, factories, and such like.
Next, I will present the methodology behind our project. We focused on COOA, a carbon monoxide-dependent transcriptional activator, as the most important carbon monoxide receptor in the detector. COOA contains heme and activates COOA-dependent promoters when it binds to CO. The COOA activator is found in rhodospirillum rubrum, but it also works as a transcriptional activator even in E. coli cells. Therefore, we designed this plasmid. Basically, when CO is present in surroundings, COOA binds to the CO. Since there is a gene for luciferase downstream of PCOM, we can add luciferin to the culture medium to get a visible light response. In other words, with the E. coli we are designing, we can know the presence of CO by emitting light. Nevertheless, to use it as a CO detector, it is critical to adjust the CO sensitivity and make the light emit at a certain CO concentration. We believe that this can be achieved by using promoters with different strengths and RBS. For instance, there are two types of COOA-dependent promoters: a strong promoter, PCOOF, and a weak promoter, PCOOM. In addition, since there are also two different RBS sequences, the results will be the following four different patterns: RBS first sequence with strong promoter, RBS first sequence with weak promoter, RBS second sequence with strong promoter, RBS second sequence with weak promoter. Due to COVID-19, we were only able to start our experiments just by the end of August. Even though we have conducted transformations with multiple conditions in a short period, the results could not be confirmed over electrophoresis. Nevertheless, we observed the color change of the medium with E. coli and luciferin under the condition of carbon monoxide. There was no noticeable color change in the medium with non-engineered E. coli nor the medium without luciferin. Thus, we assume that the system we created successfully detected carbon monoxide. Regretfully, since our lab does not own an appropriate device for observing bioluminescence, we could not measure the intensity of light. Besides, we could not observe the luminescence of the color change sample even in the dark. To solve this problem, we proposed the implementation of nanolanterns, which enable us to observe its luminescence with our own eyes. Once we obtain an appropriate device, we will also verify the luminescence intensity using various promoters and RBS.
Modeling is essential for the implementation of living organisms as gas sensors. By considering the relationship between the chemical reactions that take place in living organisms and the luminescence of luciferase, we came up with the following model. We know that the differential equation of the luciferase depends on the concentration of COOA bound to CO. By solving this equation, we get the following. In a steady state, the relationship between COOA and CO-bound substances is shown in the graph below. In addition, synthetic organisms also express proteins that are not originally produced by the organisms. This allows the organisms to work adequately as sensors. Moreover, the gases detected by this sensor are harmful. The growth rate direction rate due to CO and COOA luciferase are expressed in the equations F and Z respectively. Here, to simplify the equation, we considered a new parameter Z as the combined value of luciferase and COOA concentrations. Solving this equation and graphing it, we obtain the following 3D model. Through this 3D model, we can visually observe the balance between gene expression and sensor function as a gas sensor.
We interviewed companies related to our project in order to put our gas sensors into practice. They are Microbiofactory Inc., which uses biological devices to produce organic materials, aiming for sustainable manufacturing. Figaro Engineering Inc., the leading company of the gas sensors business. Nakura Ranch Inc., which produces Japanese beef and is a potential user of our gas sensors. The problem that we found out from the interviews was that the sensor using transcriptional control has a slow reaction rate, and that the signal from luciferase is too weak to be detected. We also learned that the current main market for CO gas sensors is in the United States. In Japan, the market is not very large, since there is currently no obligation to install them. In the beginning, we were thinking of installing sensors in smoking areas, but found that there was no demand for such areas because they were constantly ventilated. Instead, we found an increasing demand for detecting toxic gases emitted from livestock, such as cows. Future prospects include increasing the number of toxic gases that our gas sensors can detect. We also plan to develop an application that can easily detect light emission using a smartphone. Plus, to meet the sensor standards, we will work on designing devices that can withstand external factors.
The Qdai JM109 strain that we are using is derived from the Qdai K-Travis strain, which is in the risk group. Since the algae-1 strain is not associated with disease in healthy adults and has no active virulence factors, we are sure that it poses minimal health risk if taken out of the laboratory. This bacterium is designed to detect carbon monoxide. We introduced the carbon monoxide sensor protein COOA and the photoprotein luciferase into Qdai. The expression of these genes is not expected to cause any harm to humans. Research activities have been carried out using standard experimental protocols. All of members took the genetic modification experiment education and training course before using the experiment to learn about the roles and risks associated with biocontainment. Our lab practice is pipette tips and petri dishes contaminated with Qdai were sterilized in autoclave bags before disposal. When using the autoclave, care was taken to never open it until the internal temperature dropped before 80 degrees. Hand disinfection was performed frequently. Reagents that affect health, such as green fries, were handled with gloves. Since our project involves the use of carbon monoxide, a toxic, inflammable, combustible, high-pressure gas, we must be very careful in its use. In addition, proper storage and management of carbon monoxide is required to prevent it from being misused for criminal purposes. We made a chemical risk assessment before using CO to consider the risk associated with its use and consider the measurements to reduce risk. We were able to significantly reduce the risk of inhaling CO by handling it in the draft. Kyushu University has a Chemical Services Management System, IASO, which ensures that inventory and usage rates are thoroughly monitored. In this way, carbon monoxide was separated from other chemicals and stored in a lockable cabinet.
This year, Qdai succeeded in designing a plasmid for e-coil that emits light when detecting carbon monoxide. Through the art of light and e-coil, we were able to further promote the understanding and spread of synthetic biology. On the other hand, we have discovered that there are still many issues to address in order to spread our sensors to the world. In order for us to move to the next stage, we need to do the following two things. First, the detection method should be based on color change, not just light emission. Second, the detection of gases other than carbon monoxide should be possible. Through human practice, we thought about how to implement our technology into society. One of the problems we encountered was that bioluminescence is difficult to see in broad daylight. Its use as a sensor should extend to various places, not just dark ones. In addition to carbon monoxide, there are other poisonous and harmful gases in our daily lives. By making these gases detectable and allowing users to customize the type of gases detected, Qdai will be able to meet the demands of a large number of customers. As you can see, Qdai's project still has a lot of potential and can be further developed in the future through ongoing activities. This year, we were able to lay the foundation for future development. We would like to thank all the people we have interacted with in the project, and with our sponsors.
We are iGEM Qdai.